جاهزين؟
دكتور ابدأ.
والريكوردينج ضامنيه غير؟
دكتور تفضل.
ولا تحتاج أن أعمل واحد كوهوست عشان يقوم بالتسجيل؟
دكتور هو عم بيسجل مبين عنا انه اتس ريكوردينج.
تمام. السلام عليكم. Good morning. Today we will have the third lecture in the CNS1 module regarding viral and fungal meningitis. You remember we have spent the last two lectures regarding bacterial meningitis. Today we'll speak about viral and fungal. This lecture will be included in the mid-exam, because in the mid-exam you will have only one lecture in microbiology among the CNS to be included in the mid-exam. The previous two lectures, the bacterial meningitis, will be included in the final examination. So I would like just to clarify this point to you. So today's lecture will be included in the mid-exam, plus the lab, plus the small group discussion. So starting with this lecture.
دكتور تسمح لي بالملاحظة؟
نعم.
هو الصراحة دكتور يعني مادة الفاينل هيك كل المواد يعني صارت كثير كبيرة. فاحنا امس استشرنا على جروب انا وزملائي انه بدنا يعني هالمحاضرتين الاولى هي اللي تدخل بالميد اكزام، وخلي المحاضرة هاي فاينل اذا بيصير، ما بعرف هذا.
انا هذه
لانه هيك مادة الفاينل صارت كثير كبيرة، كل المواد كل مادة اكثر من 3/4 صار فاينل.
هذه you can discuss it مع المنسقة دكتورة ليلى بهذا الخصوص، لأنه اللي إجاني أنه المحاضرة الثالثة هي الداخلة، وممثلين الطلبة تبعكم اتصلوا فيا وصار الاتفاق بهذا الشأن، محاضرة واحدة على الميدل إكزام، على الميد إكزام، هذا اللي صار. فإذا بدكم مناقشة أنتم تناقشوا مع المنسقة مع ممثلي الطلبة واتفقوا على رأي، وأنا يعني ما بتفرق بالنسبة لي.
يعني دكتور مثلا لو بدنا ندخل المحاضرتين الأولى.
Please let us start our lecture. There is no time for this discussion. If you have some points, you can come to my office or ring me, please. Okay? So let us start with our lecture today. Viral and fungal meningitis. We said we are going to describe viruses from morphology, physical properties, pathogenesis, laboratory diagnosis, and the treatment of what? Of Coxsackie viruses, ecoviruses, enteroviruses, arboviruses, and rabies viruses. Remember, these are the viruses that we would like to study them today in regard to the CNS1. Also to describe a fungus, which is Cryptococcus neoformis, morphology, importance, cultural characteristics, pathogenesis, laboratory diagnosis, and a few lines about treatment. So let us start with the viruses. Coxsackie, Eco, and Entero. Very important viruses. Widely spread. They can affect the meninges, cerebellum, do not affect the motor neurons. Remember this point, very important. So these viruses affect meninges and cerebellum, but not motor neurons. Infections related to certain subgroups and serotypes, not all of them. These viruses have many subgroups, many serotypes. So some of them are important in causing these diseases. Manifestations. We are talking, remember, about meningitis. And we said from last lecture that viral meningitis is called aseptic meningitis. And we have agreed on this point. Aseptic meningitis means viral meningitis. Frequently, enteroviruses are common in aseptic meningitis. Such meningitis is usually mild, self-limiting. Within 1 to 2 weeks, it ends. It may be accompanied by encephalitis. Encephalitis is the inflammation of the brain parenchyma. Remember, meningitis is inflammation of the meninges, while encephalitis, the brain parenchyma inflammation. The most sensitive and specific test to check for these viruses, real-time real-time PCR, especially when we take a sample from the CSF. So the best diagnostic method nowadays available, real-time PCR of the CSF sample. Enteroviruses, we said the most frequent one, has seasonal peak, especially in winter, for example. They are transmitted fecal oral route. Remember, they are resistant to 70% alcohol. Some nurses, some people think that when you put alcohol on your hand or on the benches, on the tables, it will kill the microbes. Look, to this virus is resistant to 70% alcohol. One of the strains Enterovirus 71 can result in paralytic infection. And we will come to this strain later on with the CNS2. Let us take case study, and we said always we have to think about these case studies. A 2 years old girl spent a holiday with her grandparents. She developed irritability, vomiting, low-grade fever, a frontal headache over 2 days. Physical examination, she has neck stiffness. That is to say, you cannot flex the neck. Such a picture, when you hear neck stiffness or stiff neck, it may ring a bell to tell you this is a case of meningitis. Neck stiffness is one of the signs, important signs of meningitis. So, as if the history is telling me that this patient is having meningitis. Lumbar puncture, you remember lumbar puncture last lecture? I said, look to the cells. The cells are 90 cells per cubic, mainly mononuclear, and we said mononuclear, it goes with viral. Glucose is almost normal, protein is almost normal, and the gram stain negative for bacteria. Look to the phrase, negative for bacteria. So if we take this table, the same table that we have mentioned it last time, acute bacterial meningitis, we have mentioned this. Now viral meningitis, which is a clear normal or moderate increase in the pressure, white BC between 5 and 300, and today we have 90, mainly mononuclear. Mononuclear, it means lymphocyte. Protein normal, glucose normal, and the chloride is normal. So most probably this is viral or aseptic meningitis. And I have explained the other to you, so you should be aware about them also. Let us speak about arboviruses. Arboviruses, the word arbo, it comes from the following. From arthropod, we take IR, born from BO, arbo, born zoonotic viruses. So arthropod-borne zoonotic viruses are transmitted by insects, blood-sucking insects, such as mosquito. تعرفوا الموسكيتو البعوض يمتص الدم من الإنسان، وكذلك التكس sandfly, phlebotomous, right? Sandfly, we have mentioned it previously when we have talked about leishmaniasis. This is the picture of a phlebotomous. Anyway, insects, blood-sucking insects. The genera of arboviruses are four genera. Toga viruses, mainly we will talk about alpha viruses, important in a human being, flaviviruses, bunyaviruses, and reoviruses. We have talked about rotavirus previously. So today we will explain Toga, Flavi, and Banya viruses, starting with the alpha virus, which is Toga virus. This is a positive sense RNA virus, single-stranded. It is encapsulated into icosahedral capsid, the C protein. This is the C protein. It has a nucleocapsid. And then it has the viral envelope. This is the viral membrane. And then spikes, alborozat. The spikes are E1 and E2. These are protein spikes with the external diameter about 70 nanometer. These spikes, in particular, the E1 has the capability of hemagglutination and fusion with the cell membrane of erythrocyte. E2 also participate in the binding, but mainly E1 is the main spike which is going to fuse and bind with the RBC and cause heme agglutination. Virion structure, virion, it means the complete virus structure. Virion complete, flavivirus. We have two types, extracellular and intracellular, when the virus is present extracellularly or intracellularly, and we are going to see the difference between them. These are positive sense again, single-stranded RNA viruses, icosahedral capsid, lipid bilayer, this is lipid envelope, and have spikes, which is called the E diamer, okay? And also these viruses have got other types of antigens. The M antigen, which is part of the envelope membrane, M protein, and the precursor M. PR, it means a precursor. From the precursor M, the M will be formed. This is when it is extracellular. But when it is intracellular, no M, only the precursor M and the E. This is one of the differences between extracellular and intracellular. When the virus is intracellular, the E is present as monomer, the E antigen monomer. But the E antigen here, when it is extracellular, is dimer. Okay, dimer. So two differences between extracellular and intracellular. The third virus, Banya viruses, the virion structure, again, single-stranded, negative sense RNA. Now, negative sense, and you know the difference between negative sense and positive sense RNA. The positive sense, just to remind you, it can be copied directly. The negative sense, it has been to change to DNA and then copied to produce the transcription. So the virion here it is the largest one, 90 to 100 nanometer in diameter. The previous one is the flavivirus is the smallest, 40 to 50, while the previous one, which is the alpha virus, is about 70 nanometer. So the alpha virus is in between the flavivirus and the Banya virus. Also, this virus has three types of a nucleocapsid: small, medium, and large. And this nucleocapsid has got enzymes, RNA dependent RNA polymerase, which is called L protein. So you have to differentiate between L protein and L in nucleocapsid. It's a bit confusing. L protein, it means the polymerase, which is RNA dependent RNA polymerase. Also, this virus has got two glycoprotein among the envelope, G1 protein and G2 protein. This is the structure of the Banya viruses. So you have to be aware about the picture, the figures of these viruses. I would like you to differentiate between them. Concentrate on the differentiation between these viruses. Pathogenesis, a human infected by what? By the infection by the arthropods. Then you will get the virus in the blood, which is called viremia. These viruses then replicate in the reticuloendothelial system and endothelium of our blood vessels. They lead to cell necrosis and inflammation, which result in fever. These viruses have the capability of crossing a brain-blood barrier, or they can migrate along the neural pathway. This can result in aseptic meningitis, and our topic today is aseptic meningitis, and also can result in neural dysfunction when it infects the brain parenchyma and cause encephalitis. So these viruses are capable in causing aseptic meningitis and encephalitis because they are linked together. Sometimes the patient develop, first of all, meningitis and then develop encephalitis over it. So you will get meningeal and perivascular mononuclear cell infiltrate and degeneration of the neurons. Immunity antibody response usually appear after 3 days of infection. The antibodies, remember, in case of viruses, we talk about hemagglutination, hemagglutinine, and a neutralizing antibody. I will explain them to you. Hemagglutination inhibition antibody, these viruses, the name of the antibodies can cause hemagglutination inhibition antibody. There is a test called hemagglutination inhibition test to test these hemagglutinine antibodies. The test is called hemagglutination inhibition antibody. So sometimes the antibodies are named with the name of the test called hemagglutination inhibition antibody, or you can say these are hemagglutinine antibodies. The second type of antibody is a neutralizing antibody, most specific. A neutralizing antibody to catch the receptor of the virus, which is going to bind with the host cells and enter into the body cells. Today you are hearing in case of COVID-19 about a neutralizing antibody. Most of the viruses can cause the production of neutralizing antibodies. These neutralizing antibodies can prevent the entrance of viruses into body cells. As you know, IgM is the first antibody which appear against the viruses. It appeared for the first 2 months. Humoral and cellular immunity can share together in producing the specific and permanent immunity. Of course, cellular immunity will share because the viruses are intracellular. Once the virus goes inside the cells, the cell-mediated immunity will start to work. The cell-mediated immunity mainly mediated by CD8 positive cells. CD8 positive cells, which are cytotoxic T cells, in collaboration with CD4 positive cells. Treatment and the prevention, as you know, there is no specific treatment against viral infection. So supportive therapy, prevention or protection from arthropods bite, and vector control. vector يعني ناقلة المرض اللي هي الارثروبود insects. ريبيز فيروس، أجلكم الله. الريبيز فيروس يعني فيروس داء الكلب الذي ينتقل بعضة الكلاب. Envelope virus, single stranded RNA virus. It has bullet-shaped virus, similar to the bullet shape. شبيهة بالطلقة بالضبط. شوفوا bullet-shaped virus. This virus has got knob-like envelope glycoprotein, can stimulate neutralizing and hemagglutination antibodies, as we have mentioned. The virus travels through from the bites of the dogs, usually bite of dogs, sometimes bat يعني الخفاش, to the CNS along the nerves. The virus G protein binds to acetylcholine or neural cell adhesion molecule, NCAM, neural cell adhesion molecule receptor on the cells. So the virus G protein can bind to acetylcholine and NCAM on the cells of the body. This virus is negative sense RNA virus, internalized and replicate in the cytoplasm. G protein containing lipoprotein envelope is acquired from the plasma membrane. احنا دائما نتكلم على الانفلوب. الانفلوب الفيروس من يخرج من الخلية ياخذ جزء من الممبرين يصير envelope. فإذا the G protein containing lipoprotein envelope, which is acquired from the plasma membrane of the infected cells. So this is the virus, electromicrograph, bullet shape, external surfaces have a spike-like, these are the spikes, البروزات, a glycoprotein projection that bind specifically to the cellular receptors. Epidemiology and pathogenesis of rabies, داء الكلب. Two epizootic forms. Urban, الحضري, داخل المدن. Usually it comes through bite from unimmunized dogs or cats, and as I said, sometimes bats. Sylvatic البرية, wild bat, foxes, raccoon, this is the raccoon. Skunks, this is the skunk. Wolves at the app. This is rarely we encounter it nowadays. It's in America, maybe encountered in certain jobs, but usually we encounter here the urban types, which is usually due to bites of dogs, rarely from cat. The virus initially replicates in the muscles, مكان العضة. هذا الدوغ bite here. okay, في العضلة مثلا. So replicate initially in the muscle at the site of the bite and then enters the peripheral nervous system. It migrates along the nerves من خلال النيرف يصعد إلى البرين, spread to the CNS and replicate exclusively in the gray matter. The gray matter, as you know, in the brain is the outer part of the brain. In the spinal cord, the reverse. The gray matter will be inside and the white will be out. You know this from your anatomy and histology. Then, after they reach to the CNS, to the brain, they will descend back centrifugally. يعني تنزل نزول passes centrifugally along autonomic nerves. يعني ينزل من البرين من خلال الاوتونوميك nerves to different tissues including salivary glands, adrenal medulla, kidneys, eyes, lungs. يعني ينزل الفيروس من البرين على هذه التشيوز. The presence of the virus in the salivary gland of animal also facilitate in the further transmission of the disease. يعني أجلكم الله الكلاب تحمل الفيروس in the salivary glands. So we can check the animal also. We can catch certain groups, can catch the animal, bring it to the laboratory, and we can check the dog, for example, whether it carries the virus or not. And I will talk about this in a moment. So as we said, the virus inoculated, replicate in the muscles, the virion enters the peripheral nerves, a passive ascend via sensory fibers, replicate in the dorsal ganglia. This is the dorsal ganglia. Then it goes to the spinal cord, ascend in the spinal cord, and then reach to the brain stem, cerebellum, and other brain structure. And then descend via the autonomic nervous system to different tissues, salivary gland, eyes, adrenal medulla, and other tissues, of course.
دكتور
نعم. إذا نخلي سؤالك بالنهاية.
ماشي.
Now, how we know, how we detect that the cells are infected? There is what is called negri or inclusion body found in the neuron. This is pathognomonic. يعني شنو pathognomonic? Diagnostic. This is negri body. Negri body in the cytoplasm of a neuron. هذا هو النجري body. هذا هو النجري body. Okay? This is pathognomonic. Once you see it in a neuron cell, you say this is rabies infection. The incubation period is variable, very variable. واحد من يعض أجلكم الله الكلب, it can goes into very long incubation period up to 1 year, from 10 days to up to 1 year. The average between 20 and 90 days. If you immunize the patient or the individual who has been bitten by a dog during the incubation period, this immunization will abort the infection. If signs and symptoms started, you cannot abort the infection at all, and the patient most probably will die. So very important to do immunization during the incubation period to abort the infection. Rabies passes into 4 clinical stages, and we will see them in a moment. Encephalitis is common, and sometimes with ascending paralysis. Once the symptoms started, the disease will be fatal. What are the 4 stages of rabies? Look, this is the incubation period. We said it varies from 10 days to 1 year, average between 20 and 90 days. هاي البقية ما أريدها منكم الهون، فقط الستيجز. Prodrome stage, it takes about 2 to 10 days. Acute neurologic stage, 2 to 7 days. Then coma, which usually the patient may immediately goes on coma and then die, or may stay in coma up to 14 days. Unfortunately, once these symptoms started, the last stage will be death of the patient. So with the incubation period, there will be 4 stages: prodrome, acute neurologic stage, coma, and death. A clinical capsule, and we usually concentrate on the clinical capsule. Rabies involve development of severe neurologic symptoms and signs in the patient. The neurological manifestation characterized with restlessness. المريض لا يستقر. Agitated. كلش منثار. Hallucination. يتهيأ له أشياء. Salivation. Very important salivation among the mouth. Foamy mouth. حتى كأنما في روة على الفم. The patient appear to be foaming. Foaming شو يعني معناتها؟ معناتها في رغوة على الفم. And have severe throat contraction if swallowing is attempted. إذا تعطيه شوية قطعة في فمه to be swallowed, cannot, will be followed by severe contraction. Neurologic abnormalities are explained by the spread of the virus from the bite where the wound is present to the CNS and then descend back through the autonomic nervous system to different tissues. So this can be related to the clinical picture that appear. Where are the viruses are present? Diagnosis, we can divide it for the patient first and for the animal. As I said in the beginning, for the patient, CSF, minimal abnormality in the CSF or even no abnormality. Sometimes you may get lymphocytic pleocytosis. A lymphocyte may be increased. Immunofluorescent test, direct fluorescent antibody stain, you can take nape, a small piece from the neck biopsy, which is best diagnostic test. ليه النج؟ يعني why the neck؟ ممكن واحد يسألني دكتور ليش من النج؟ لأنه once the virus will descend from the brain, أقرب tissue إلى البرين هو النج. So neck biopsy will be the best diagnostic area. PCR for viral antigen in the CSF or saliva. Antibodies can be detected late in the disease. Serum antibodies are not raised immediately. They take some time to be raised. So usually not useful in the immediate period after the biting of the animal. For the brain, to detect the virus, either we use immunofluorescent, for example, dog, or animal inoculation. We take the sample and inoculate it in mice, sucking mice. The mice will die within 3 to 10 days. We inject it intracerebral inoculation في داخل دماغ المايس. Negri bodies, as I said, found histologically in the brain tissue in 80% of the cases, Also EM microscopy, electron microscopy يعني can detect negri bodies and the rhabdovirus, which is the road particle, the bullet shaped in the brain tissue. A treatment, we can divide the treatment into pre-exposure يعني في بعض الجوبس اللي تستوجب البري فاكسينيشن مثلا بوتشرز القصابين الصيادين هانترز دول معرضين الى الدوغ بايت فممكن نلقحهم قبل اصابتهم او التلقيح ما بعد عضة الكلب post exposure. So pre-exposure and post-exposure. Rabies vaccine started by Pasteur in 18,000s يعني في 1800. It can induce neutralizing antibody and cytotoxic T cells, CD8 positive. As I mentioned, very important in viral infection. The pre-exposure to high-risk individual. Usually they are given inactivated vaccine, killed vaccine. 3 جرعة ياخذوا. Day 0, day 3, day 21 or day 28. يعني إما يوم 21 أو 28. يعني 3 أسابيع أو 4 أسابيع. ولهذا الآن تسمعوا على 3 أسابيع أو 4 أسابيع بعد الكوفيد فاكسين. البوست exposure treatment, if the animal retained to be negative, have negative test for rabies, no treatment is suggested. If the animal is positive for rabies virus, a treatment will be given. What sort of a treatment? You should wash the wound by soap and water, of course, but the main thing, passive vaccination to give antibodies, anti-rabies, hyperimmunoglobulin, single dose in day 0. With it, you give active immunization, activated vaccine in day 0, 3, 7, and 14. This is if the patient has not been immunized previously. If the patient has been immunized previously, مثل ما حكينا في بعض الجوبs like butchers أو hunters, we give only 2 doses, 0 and 3 days. Sometimes even give 1 dose, and we don't give hyperimmunoglobulin. We don't give antibodies, passive immunity, because the patient already has been vaccinated. Lastly, we talk about a fungus, a Cryptococcus neoformans, a genus of yeast, Cryptococcus, بها شبه من الكانديدا. الكانديدا is yeast. The Cryptococcus is also yeast. It has a diameter of 4 to 6 micrometer in diameter. It is capsulated. This is a difference from Candida. The capsule contains complex polysaccharide polymer. Complex polysaccharide polymer. Remember, GXM. The name of the capsule antigen or the capsule itself is called GXM. So Cryptococcus neoformans is capsulated yeast. The colonies appears in culture in 2 to 3 days at 25 degrees centigrade or 37 degrees centigrade. This fungus or yeast have got enzymes, urease enzyme and lacase. Lacase can induce melanin pigment from acetylcholine. Very important in the pathogenicity, this lacase, and I will come back to it in a moment. So urease, remember, this is urease positive. Cryptococcus neoformans, remember, it is urease positive. I repeat again, Cryptococcus neoformans is urease positive. This is a main difference from Candida. The capsule is another difference from the Candida. Clinical capsule, the primary disease of Cryptococcus can cause chronic meningitis. Remember the word chronic meningitis. That is to say, slow course, insidious, بطيء. The patient develop low-grade fever, headache, and then progress to altered mental state and seizure. حتى مرات نتيجة الالتر mental state the patient suspected to have psychological abnormality يعني psychosis وكأنما يظهر المريض على شكل أنه ما يشبه المجنون psychosis. In the cerebral spinal fluid and in tissues, the inflammatory response usually very little. There is no frank immune response. Most patients who are infected with the Cryptococcus neoformans are immune compromised. فيعني الانفكشن المننجايتس تبع الكريبتوكوكس neoformans in immune compromised. Like what? Like in AIDS. It could occur in patients without immune defect. But usually I am saying it occurs in patients who are immune compromised, like in AIDS, or patients taking corticosteroid or cytotoxic drugs, or have immune deficiency diseases. Look to this picture. This is called Indian ink preparation. الحبر الهندي حبر. When you put it, mix it with CSF containing cryptococci, they appear as yeast. هذه الدوائر surrounded by capsule. هذا الهالو around it. This is the capsule of the virus. Because it is yeast, it has budding. This is, I have arrowed it. This is the budding from the yeast cells. So remember, Indian ink, capsulated, budding. Epidemiology, this condition with the Cryptococcus neoformans infection associated with soil and bird droppings. Bird droppings يعني الستول تبع البيرد. اوكي، فالمناطق اللي تكثر فيها، يعني احنا اجرينا دراسات على هيك موضوع، شفنا انه بالمناطق اللي يكثر بها البيردز والبيرد dropping it may result in spread of Cryptococcus neoformans. As we said, this is yeast and basidiospores. These buddings, which are called basidiospores, can be inhaled by patient. And as we said, it is common in immunocompromised patients like in AIDS. Pathogenesis, the capsule is antiphagocytic. And you know the capsule usually antiphagocytic. Sometimes we can detect the capsule material which interfere with the immune system. Melanin, oxidative protection in macrophages. The melanin that we said that this microorganism can produce through the activity of the lacase provide a protection to the Cryptococcus inside macrophages. Cryptococcus neoformans can cross the blood-brain barrier in macrophages. So the macrophages represent the Trojan carrier, a Trojan horse. يعني حصان طروادة, Trojan horse, وكأنما الماكروفاجز تنقل الكريبتوكوكس للبرين. Clinical manifestations, slow, as I said, insidious onsets with non-specific findings, intermittent headache, irritability, dizziness, difficulty with complex cerebral function ما يقدر يفكر, and behavior changes, حتى differential diagnosis, psychosis, الجنون. The patient may have seizure, cranial nerve signs, and papilledema. This is papilledema, look at it. That is to say, optic disc swelling. نشوفه by retinal examination. Papilledema means optic disc swelling. Diagnosis: CSF, increase of pressure. lymphocyte، لو ترجعوا للجدول هلا راح تشوفون مطابق لما مذكور هون. lymphocyte pleocytosis, more than 100 cells. Decreased glucose, capsule stain Indian ink, 50% positive. Culture, you require a lot of CSF, more than 30 mil to culture, because the cryptococcus are very scanty organism in the CSF. GXM polysaccharide can be detected in CSF or in the serum, as I said. And antigen titer has got a prognostic value. يعني antigen titer يعني قديش عندنا من البوليساكرايد بالسي اس اف او بالسيرم. PCR is very important diagnostic test for detection of Cryptococcus neoformans in the CSF. A treatment, the last slide, fluconazole. 75% respond properly. Relapse, significant percent, which require to repeat the course. 50% of the cured will have residual in neurological damage. Okay, so by this we end the lecture, but just to remind you with the fungal meningitis, which is a Cryptococcus neoformans, a clear CSF, pressure increased, white BC more than 100, mixed, protein increased, glucose decreased, chloride is decreased. Very important to know this profile. This is the profile of fungal meningitis. An example of fungal meningitis is meningitis caused by a Cryptococcus neoformans. Any question?
دكتور
نعم.
لما كان البايت تبع المصل، لما يعمل
نعم.
الفيروس ريبليكيشن جوا المصل، هو بيعمل انفكشن بالمصل ولا بس يعني بيعمل ريبليكيشن؟
يعمل بالمسلز، يعمل بالمسلز انفكشن.
يعني بيعمل اول شيء انفكشن بالمصل بعدين
مذكورة لكم replicate initially in the muscles. هتشوفين السلايد. Replicate initially in the muscles at the site of entry and then goes through the peripheral nerves.
يعني يمكن يصير انفكشن اول شيء بالمصل بعدين بالسي ان اس.
initially تعتمد على يعني المنطقة اللي تلوثت فيها، أكيد في infection لأنه initially replicate in the muscles and then it travels upward. ولهذا السبب بالتريتمنت لو تلاحظي الخطوة الأولى اللي عملناها بالتريتمنت you wash the wound with soap and water just to clear the area.
تمام دكتور.
تمام. Another question? اوكي. طالب اللي سأل على المحاضرة وعلى هذه ممكن تنسقوا بيناتكم وتطلبوا من المنسق، بس أعتقد أنه محاضرة واحدة تدخل عندكم بالميد إكزام أفضل، لأنه محاضرة واحدة وليست محاضرتين، يعني تخفف عليكم العبء، يعني هذا المقصود يعني كان فيها. لأنه ذيك محاضرتين مرتبطة مع بعضها. حصة الميكروبيولوجي بالميد إكزام 1 lecture only، لأنه في لاب وفي small group discussion، فحتى يصير توازن فمحاضرة واحدة لا بيجيكم من عنده مايكروبيولوجي لا بقصد وسمول جروب ديسكشن، فيصير توازن. بالفاينل اكزام only 2 lectures، there is no lab no small group discussion، فيصير توازن يعني. فأعتقد أنه هذا الخيار يعني متوازن بالنسبة لكم. Other comments؟ If not أتمنى لكم التوفيق إن شاء الله في هذا الموديول، ونلتقيكم إن شاء الله في السمول جروب ديسكشن أو في الموديول القادم بإذن الله. السلام عليكم.